Welcome to Inline Automation and Miniaturized Reaction Volumes for Real-Time Isothermal DNA Amplification. I'm Tamlin Oliver, Managing Editor of Genetic Engineering and Biotechnology News, and I'll be the moderator for this webinar. I suspect, based on the fact that you've signed up for and are listening to this webinar, that you're very familiar with PCR, and I don't need to go into the pain points and frustrations inherent with the multiple instruments and numerous human contact points involved in real-time PCR processing. I will instead let our three speakers tell you about a new way to get around the frustration and go from sample to answer in 20 minutes or less. Sound too good to be true? Dan Schaefer, Manager of Technology Development at EnviroLogix, Craig Vinci, Global Sales Director at Douglas Scientific, and Cassie Keppel, Manager of Laboratory Automations or Operations at Douglas Scientific, say their method reduces the time required for amplification and real-time data generation, increasing throughput and decreasing cost per data point. I'll let them give you the details in a minute. Dan Schaefer will kick off the webinar by talking about EnviroLogix DNable chemistries. Before Dan starts, though, I want to encourage you to submit questions for our Q&A session at the end of the presentations. We'll answer as many questions as we can. Simply type your question into the Ask a Question box and hit Submit. Dan, we're ready for you to get started.
Thank you, Tamlin. I also want to thank Genetic Engineering News in general and my colleagues at Douglas Scientific. I also want to encourage you guys to listen all the way through to the very end, because there's some very interesting new data fresh off the presses from Cassie Keppel that basically shows you the true sort of synergy and combination of the Douglas Scientific array tape system with the DNable system. And with that said, what I'm going to cover today is the EnviroLogix interaction with Douglas Scientific, a real short little description, and then go through some of the chemistries associated with this type of isothermal amplification system that we call DNable. There's a version 1, and then we're going to talk about the version 2 chemistries and how that has improved the system, and then, of course, talk a bit about the high-throughput assay systems associated with the Douglas system. And it's exciting for me because, to be honest with you, about 1,000 years ago, before my beard turned gray, I was a field application specialist for Applied Biosystems when they first rolled out the TACMan system, 7700, etc. And of course, that was game-changing. That was disruptive. That was exciting. And I want to just sort of bring to you the same passion and excitement about this process of linking an isothermal system with the Douglas system. One of the things that we want to be clear about is that EnviroLogix has this nice collaboration with Douglas Scientific that enables us to bring to market a complete solution that enables real-time and endpoint nucleic acid analysis. So again, by combining the DNable chemistry of EnviroLogix with the array tape and inline liquid handling and sample processing systems of Douglas Scientific that both Craig and Cassie will talk about in detail, it provides an unprecedented and cost-effective capability and throughput with regard to amplification and detection systems. It's exciting for me, and I want to try to drive forward that same sort of energy and passion. But since I'm not going to be really talking about qPCR, what I want to talk about, in essence, is basically how the gold standard of qPCR has been established in the past, and how isothermal has, in all honesty, kind of had a tainted reputation with regard to its performance as compared to qPCR. Now, qPCR shown here is classic PCR, primer-driven, etc. And what I want to flag here on the right is the panel of amplification curves in the top portion of that right panel. And those are beautiful sigmoidal amplification curves showing a dilution series, and below that is the typical standard curve that everybody expects to see from qPCR. But I want you to just keep those images in mind, because as I go forward and show you things associated with DNable, that's what I want you to keep in mind, and show you how we have established a system with an isothermal world that reflects the same sort of gold standard that you've come to expect with qPCR. Now, with that said, way back in the early days when I worked for Applied Biosystems, this is one of the slides that indicated to people that it was really important to collect things in real time, because there's a stochastic or random event that causes the reaction to basically fall out of log phase and end up with a variable endpoint. And those variable endpoints are shown in that red oval to the right, where these are 96 technical replicates that were all generated on the same block at the same time, and you can see that they have a lot of variability at endpoint. However, down low where they first begin to show the log phase amplification in the little green oval, things are more reproducible there. So, of course, in real-time PCR, you're collecting all of the information simultaneously as the reaction is occurring, whereas in other applications, you're collecting at endpoint. So you may not necessarily collect it as real-time, but you still collect it at endpoint, and those signals can vary. And that's classic in typical qPCR and endpoint-based PCR with fluorescent-type detection. But I want to bring to the table here the de-enable reaction, because it's something that is basically really simple. I mean, if you are familiar with qPCR reaction components, you have a DNA or RNA target. The RNA may be converted to cDNA. We typically have two primer-specific or target-specific primers. You have your dNTPs and you have your DNA polymerase. Now, those are the familiar components. In the context of the de-enable reaction, there are components that may not be as familiar to you unless you're familiar with many of the isothermal systems that are out there. So in the case of the de-enable system, not only do we have the components listed above, but we also include a nicking enzyme. An amplification, in this case, that occurs in typically 10 minutes or less. And then, of course, target DNA that does not necessarily need to be isothermal. It can be purified. And then, of course, the system is isothermal. So those are the things that are not necessarily quite so familiar to people. But let me just roll through the reaction to bring you up to speed on this system. Now, I have a series of cartoons here that I'm going to describe. And now, suffice to say that this is rather simplified, but it brings across the point that I think is going to be quite simple to understand. First of all, looking at the top right, with that picture there, in essence, you're looking at double-stranded DNA. And throughout the double-stranded DNA, of course, you have restriction-like sites. And in this case, there are sequences distributed throughout the genome to which our nicking enzyme recognizes. In this case, it's a BST derivative. So it's NT-BST NB1 nicking enzyme. And that nicking enzyme binds to a sequence that's GAGTC, 5-base recognition sequence, and then it nicks that same upper strand that it was binding to about 4 bases downstream. So it does that throughout the genome. And that little second portion down below where you see the 3' and the 5', just downstream of the little box, which is the nicking recognition sequence, it generates a little nick in that position. And then the DNA polymerase binds to that 3' end and progresses and generates a replicate DNA strand while simultaneously displacing the upper strand. So it doesn't cleave the DNA as it progresses downstream. It displaces it. And it does this over and over and over again as it regenerates the nicking enzyme recognition site and nicking site. Again, it can serve as an additional nick displace position there and generate lots of single-stranded DNA, kind of like what happens in PCR when you melt the DNA at higher temperature. So in essence, this gives you that first step, that first denaturation step, generating lots of single-stranded DNA available for the next step. So shown here is the next step. What we do actually in this reaction is we have, in this homogeneous system, the primers that are actually engineered. The primers have a recognition region on their 3' end that is specific to the target sequence of interest. And then additionally, there are sequences added onto the 5' tail that includes that same nicking recognition sequence, such that as both primers begin to anneal and extend, you generate a product that's shown down to the bottom that includes that product of interest, but you've now engineered and included a new restriction enzyme site there, nicking enzyme site, that is now a substrate for the NT-BST-NB1 enzyme to nick. And then, of course, the polymerase binds and displaces, and in this cartoon you can see all the way down to the bottom, you begin to generate that strand-specific, or excuse me, the target-specific single strands that are available for detection. And this is basically what we call kind of the nick and kick. So as you've generated that product, you get a nick, you get the displacement, and it kicks off the upper strand, and then it regenerates more and more and more. So you get this logarithmic amplification phase at this point. And what we use is a molecular beacon, which is similar to a TACMAN probe, although it's a stem loop structure that binds to that specific product, since it's a single-stranded product. Then, of course, the fluorescence is generated by a molecular beacon, and, of course, detected in a typical detection system like a qPCR instrumentation or the instrumentation that you're going to hear about from Douglas Scientific. So in a nutshell, that's the reaction. Now, there's a lot more to it than that, but that's kind of just to familiarize yourself with it. But I want to talk about de-enable version 2, because what I showed you is the classic and clean cartoon of the world. The reality of it is the fact that it is known that isothermal systems cause and create lots of background products. So when we first took a look at this reaction system, we were challenged with coming up with conditions that did not generate these background products. So there's non-specific amplification products that are kind of akin to primer dimers and PCR, and there are challenges to minimize those. And what I want to suggest to you is that in the challenges to eliminate those, that would also mean that if we eliminate them, we have to at least have the same sort of performance that we had prior in the DNA DNA-enabled version 1 chemistry. And with that said, if we can control the reaction so that you're making only specific products, not these background products, primer dimers, etc., that should enable you to do the classic things that you would expect to see in a qPCR reaction, such as duplex or multiplexing, etc. And also, just want to point out at the very bottom of this list, previously the enzymology was such that we would actually have to set things up in what was considered a mock hot start. In other words, we actually built the reactions while they were up at temperature. And if we can control the reactions in a way that is mimicking their typical tack gold or something similar to that kind of methodology, that should enable us to do what we consider like a cold start. Basically set it up on your bench or on a liquid handling robotics workstation and have the reactions sit there and not go until they actually are brought up to the appropriate temperature. So everything that we're talking about here was basically developed over the past year or two that enabled us to take the original DNA-enabled system and convert it to a version 2 system with some very simple chemistry changes and dramatically improve the performance of the reaction. And here's an example of that performance change where the left panel is the version 1 and the right panel is the version 2 results, looking at the amplification of a particular pathogen. And in this case, we're detecting via cyber green, which detects any sort of double-stranded DNA. And on the left panel, you can see that in this case, we've got a dilution series of the targets, and there's really not much dynamic range there. It's rather compact, although the reactions happen very quickly and very nicely. There's also, shown by the red arrows, a problem, and that is that the no-target controls give you a lot of signal, generating or demonstrating that there is a lot of junk that's generated in the background. But if you convert that system, literally exactly the same system, change out the chemistry slightly, you can now convert that kind of reaction into what really is now behaving like your classic sigmoidal amplification curves that you see on the right panel that you would expect in a qPCR world. But more importantly, the red lines that are flat are the no-target controls. So we've now shown that we can control the noise generation in the background, and therefore we get a much, much better kinetics for the specific target being generated. Now, this is using cyber green, which is, it's detecting any junk that's in there. If I then take exactly the same reactions and simply detect them by molecular beacon, the left panel shows that at the higher concentrations you get a very nice amplification curve, but as you begin to dilute the target down, you begin to lose efficiency because it can't outcompete the background products that are also in the background being generated in the version 1 chemistry. But if you convert it to the version 2 chemistry, you can see you get back to that beautiful log phase, matched efficiencies, sigmoidal structure. Really looks like a typical qPCR reaction and behaves the same way. But everybody knows that in order to do this, they want to see what's really going on in the reaction. So we took those NTCs directly from that experiment I just showed you and ran those individual ones, in this case, we were showing 3 NTCs, on a mass spec. And on the left, you see a series of 3 individual reactions that were derived from that experiment that are, again, those NTCs. And the background components that are shown here shows a very complex spectrum. So you're looking at multiple mass-to-charge ratio entities in this system here. And then by converting it to the version 2 chemistry, you eliminate those, shown by a very simplistic or simplified non-complex spectrum on the right side. So it just further drives home the message that we've cleaned up the system with the version 2 chemistry. But the challenges of this system is to try to demonstrate that the capabilities of isothermal is as good as the gold standard with PCR or qPCR. And one of those things that people always challenge you about is, well, great, what's your standard curve look like? What are your efficiencies? What's your R squared on your standard curves, et cetera? And here's just an example of and your dynamic range, of course, but of a series of dilutions of a target, in this case, showing that we have greater than 7, maybe 8 logs dynamic range, maybe even more. With very tight replicates that may not be visible, but the R squared in this case is 0.99, I believe 16. Again, it's demonstrating that the capability of the version 2 chemistry can very closely mimic the gold standard, again, though, in an isothermal world. For us, isothermal means the reaction is run constantly at 56 degrees. There is no additional temperatures added on. It's 56 degrees for a short period of time. And in this case, what I'm showing you is this dynamic range is accomplished in a 10-minute time frame. So again, that's quite significant in terms of isothermal and short, rapid turnaround time for the reactions. But a beautiful thing about this reaction is that it can really tolerate some really ugly samples. Shown on the left is a tube of what looks like green goo, and that's actually a citrus leaf that's been ground up in an extraction buffer. And literally, we took that exact same matrix, doped in some known counts of, in this case, Clavibacter michiganensis bacteria into that sample, and then took small aliquots across different dilutions, in this case 2.5 microliters per reaction in a 25 microliter reaction, and looked at these different dilutions in that kind of background. And we're just showing you the unprocessed raw data of the amplification curve shown here, where the one that's triplicates that's shown kind of down low and on the right is the equivalent of seeing or detecting 2.5 colony-forming units per reaction in a crude matrix like this, demonstrating that really this reaction system is quite tolerant of some pretty nasty matrices. But I'm going to push it even farther than that. Let me show you some other things after the fact. But the simple aspect of this is also that the protocols for doing the sample preps are extraordinarily straightforward. In the context of what I just showed you, literally it's just taking a small punch of little discs of leaf tissue, grinding them up very quickly, either with a pestle or a bead beater or whatever, in a small volume extraction buffer, in this case 200 microliters. Heat it for 5 minutes, add a dilution buffer, and then basically pull your sample out and run it. So it's a very short sample to data processing time. So, at the bottom I show here, are you missing something? Where's the DNA purification step that we've all come to kind of loathe with regard to qPCR? And in many applications, we can literally just do away with that. I'm going to show you more, because what really sets us apart is the tolerance for crude samples. Here's an example, again, of the same bacteria that was spiked into what's called a seed wash. And for any of you guys that have done work on trying to detect CMM in a large batch of seeds, the seed wash protocol is well known, and what you end up with is a pellet in the bottom of your tube that, once you dissolve it, is actually very milky or sometimes a muddy-looking sample. And we wanted to determine how well this system would detect in that seed wash type sample prep by, again, doing a spike in and a standard curve. And you can see in this case, very nice linear dynamic range, very nice R squared, et cetera, in a very challenging matrix for this bacteria. But let's make it even harder. Let's go into a world where trying to detect, for example, E. coli in cow feces. Now, we're not talking about purifying the E. coli from the cow feces. We literally spiked in these samples, in this case, E. coli, into batches of cow feces, did the extraction as we do, basically the lysis, and then basically ran the detection using an E. coli ON57 specific target. And again, we got a very nice dynamic range, et cetera, in this reaction. So again, demonstrating that the reaction is quite extraordinarily tolerant of sample prep and contaminants and things that would certainly kill PCR. Let's, again, keep pushing the sample prep aspects of things, that we can do these sorts of things in, for example, crude, in this case, soy, where we're trying to detect a GMO in the presence of a crude sample, in this case, crude soy. So we're looking here at a duplex reaction, because it's a DNAble version 2, that we can now do duplex reactions with an internal control, where the gene of interest is labeled, the beacon is labeled with a reported I called FAM, and a endogenous gene in the soy, lectin, is labeled with a beacon, detected with a beacon labeled with hex. And so you can duplex a reaction and have, of course, a positive control built in there for the presence or absence of a GMO in a crude prep. Literally just grind the soy, put it in a very, very simple buffer, a quick heat step as you saw before, and then run the reaction in the tube. Pushing the envelope further again, looking at two different targets within a particular pathogen. Duplexing is certainly doable. This is just an example of STIX1 and STIX2 in E. coli. If you're a 157 flavor or cousins, we can detect in a duplex reaction. This is actually a rather old slide, but a duplex reaction for those two in the same reaction that verifies whether or not you have a 157 or cousins or not. In addition to that, again, what I wanted to demonstrate here is the specificity. So what we're looking at in this case is a particular GMO event, in this case in a crude leaf tissue, showing the specificity for the event, in this case event 2, that shows up very nicely, and then from the same samples, not co-amplified, but from the same samples, it's negative for events 1, 3, 4, 5, and 6, the other events that could potentially be there. Again, just demonstrating, again, same sort of opportunity here, looking at similar type of systems with a different event this time, no cross-reactivity to the other events in these core tissues. So again, if you were looking for stacked events or just a single event, we have that capability with, again, a very simple crude prep and detection of events. One of the things that's of interest, of course, is SNP detection, and we can demonstrate SNP detection in this case in a synthetic example where we're looking at alcohol dehydrogenase 1 from corn sequence, where we're looking at a C to T SNP using 2 beacons, one labeled with fluorophore 1 and the other labeled with fluorophore 2, a G to A difference between the 2 beacons. Simply go in and, in this case, doped into crude corn background the specific target of interest using a G block, and identified very quickly, literally in the first pass that we did this, the detection capability of homozygotes and heterozygotes in this test system using alcohol dehydrogenase synthetic system. So again, signal-based discrimination by molecular beacon in isothermal systems is quite straightforward. But here's where it gets kind of interesting. The de-enabled detection of E. coli STIX1 in this sample and complex matrix, in this case, is showing that the system works very nice, whether it's detected in TE or whether it's detected in a crude sample with E. coli feces, with cow feces. But if I try to do the same thing with a PCR, you can see the PCR amplifies very nicely if the sample was done without the feces. The minute you put in the feces, you kill the PCR reaction. And that red line shows the 10 minutes where we had already detected both in the TE and in the cow feces by de-enable. So again, showing how quickly things can go in that regard. So the enable version 2 gives us capability of doing all of the things that I hoped to be able to do with regard to being directly in parallel with the expectations for the gold standard being qPCR, but again, minimizing the sample prep issues, and then, of course, the time to getting the results is significantly faster. But what I really want to show you is the Douglas Scientific Nexar system and the array tape system. So as we go forward with this, what I'm showing you here is the capability of dispensing in a system, in a lab deck-based or a robotic-based system, 384 well arrays. So each little batch of data, the blue batches on the bottom left and the red batches on the right, show the detection in the Nexar system using 1.6 microliter reactions, completely set up, automated, using, in this case, an ideal system, the cleanest system that we could have. And again, it's that alcohol dehydrogenase G block and 10,000 targets available in 2 3D4 well plates and the red splotches on the right, and then the no target controls on the bottom left. So that's the ideal system, but the system that would be challenging would be to do it in a matrix that looks, for example, like this, where the soy sample is shown on the right and in comparison to just plain water on the right. So taking those soy samples, or that soy sample right there, and literally dropping it into the Douglas system and running the reactions similar to the ideal system gives you results that are basically indistinguishable. Beautiful detection of, in this case, the crude soy using lectin as the target, and then, of course, the no target controls showing literally no signal down below. And then the inset on the right just shows you what the real-time PCR amplification profiles look like, showing, of course, the typical spread of endpoint signal that you would see in this or in qPCR. Throughput is everything. So with isothermal systems and the ability to set things up on a deck space like the Nexar system that you're going to hear about, it's all about throughput. And again, right here, 1.6 microliter reactions saves you reagents and, of course, time to get to your results quicker. And I just want to show you from my personal experience how this feels to me. Because in a previous life, this is a stack of 300 arrays that I had to do, 384 plates that I had to do, and I did them basically manually. It took me about 60 days to generate the data using a real-time PCR machine. But in the context of this, you can accomplish that literally in a day or two on the Douglas system and have significant savings with regard to volumes of reactions and costs. And I know that Craig's going to talk about that a bit further. So the summaries in terms of this system is that, in essence, the features are very similar to qPCR. You've got first layer of specificity by primers, second layer of specificity by beacons. You've got, again, a wide dynamic range and theoretical single molecule detection, just like qPCR, but here's where things get different, and that is the sample prep. So qPCR typically needs cleaner or even just plain purified genomic DNA. DNable tolerates crude sample preps, as I've shown you. And, of course, it can handle clean DNA, and feel free to use clean DNA, but there's sometimes options that are necessary that don't enable you to purify your DNA. We have very high signal-to-noise ratio that's compatible with the small volume reactions as shown in the Douglas system, and you'll see more. And in addition to not only the sample prep improvements, we also, of course, have very, very short time to get to the data. So the faster you can do it and the smaller the volume you can do it in, the more you can save, the more you can get done, and you can really move things forward. So the summary here is that there's a fundamental understanding of the reaction. That's enabled us to have better, faster, easier assay development. And there's predictable assay performance. We have a software application that we use called the Enabler that was developed here. And then something I didn't talk about, that there is a feature to the chemistry that we can use to tune the reactions. There wasn't time here to talk about that, but we can talk about that offline. Because of the improvements that we've had, we have improved assay performance. In other words, higher specific reaction efficiency because we're not doing the off-target amplifications. We have expanded capabilities with endogenous or exogenous internal controls. The applications are basically anything that has previously been owned by PCR, such as SNPs, presence or absence assays, absolute quantifications, relative quantifications with internal normalization, et cetera. And then, of course, segueing into the high and ultra-high throughput endpoint detection using the Douglas Scientific Nexar system, optimized, or the EI Nexar. And then, of course, what you will be hearing about is the next generation real-time detection system from Douglas Scientific. So with that said, I think the next person coming up is ready to go, and I hope that you're excited about this and you're going to see some additional things from me. So thanks for listening, and I will be here for questions later on. Craig?
So actually, Craig, before you get started, I just want to remind everyone about submitting questions through the Ask a Question box. Thank you, Dan, very much for your presentation. You certainly covered a lot of material in a very short time, and Craig does have a lot to talk about. So, Craig, we're ready when you are.
Okay. Thank you very much, Dan, for that information. And my name is Craig Vinci, and I am the Global Sales Director for Douglas Scientific. I want to thank everyone for attending today's webinar, and we're very excited about the solution that we're presenting here today. As Tamlin stated in the opening, I'll be building on the material that Dan shared and that Cassie will eventually share to show that this is the complete picture of how the isothermal amplification addresses the pain points that are associated with the current traditional PCR methods, and illustrate how together with EnviroLogix we're providing you a total solution. The inline automation that Douglas Scientific offers, I break up into 3 points. One, I'll tell you just a little bit about the company, and then the automation itself, and then go into the application. Douglas Scientific is located in central Minnesota, as you can see on the map here today, and we are a fast-growing young company, but we do have over 100 systems already in the field, and these are mostly running Endpoint PCRs. So we're really excited about what we're showing you here today, where we're running the isothermal DNA amplification through our partnership with EnviroLogix. I think it's always important to state our mission statement at Douglas Scientific, and that is making the world a better place by delivering innovative laboratory automation. And not to put too fine a point on it, but keyword there was automation. That's what we specialize in. So to really realize our mission, it's important that we make partnerships with companies like EnviroLogix who really bring the chemistry expertise so that we can realize the mission of our company. Okay, on to the automation. First, I want to talk about the evolution of laboratory automation. So doing things in large volumes and in test tubes has its problems that we're all aware of. No need to go into it here. And through the evolution, The multi-well plate, of course, was invented. But what we hear from customers is that they want to do more and more. Dan mentioned throughput, and that is critical. So as the throughput increases, as you see over in the third picture, so does the complexity of the robotics that are required to move the multi-well plates around. And so at Douglas Scientific, what we've done is taken it to the next step and gone to the inline automation, where you have a single reel that contains all of your plates, or a good portion of them, and they run continuously through the system. And what this allows you to do is do more with less. So you'll see in the next few slides how we minimize the reaction volumes. There's considerable savings there, and considerable savings not just with the consumables themselves. So we feel that's really a shift in laboratory automation. So to zero in on the array tape itself, it's a continuous polymer media, as I mentioned, and the wells are thermally embossed into the array tape. So what does it look like? The blue reel that you see sitting there is about 1/2 of a meter diameter, and that contains over 200 arrays, each with 384 wells. And what you see sitting next to it is the same number of microfiber plates. So you can see the benefits. There's a reduction in the carbon footprint. The amount of plastic to do the same work is significantly reduced in the array tape. And a clear reduction in storage space. It's a lot easier to handle where you're handling just one single reel rather than all the individual plates. The array tape is flexible, and so we can make it out of multiple different materials, different well volumes, assay-specific opacities. On the right there, you see the white array tape, and that's to be used with certain detection modes. And thermal embossing allows rapid prototyping versus injection molding. So we don't have the expense that you find with injection molding when we need to come up with a certain well geometry. So it allows us to do the rapid prototyping and come to a solution much faster. So for liquid handling, you might be wondering how you are going to fit this into a liquid handler. So Douglas Scientific has developed an inline modular liquid handler that we call the Nexar, and that's what you're looking at here. And the system starts from the right side at what is listed as the Untitled module, and the array tape is fed through the system, going right to left, first into the next section that you see is called DNA dispensing and tip wash. And what that is essentially a 384 head, and that transfers your samples that are coming from the stacker that you see immediately to the left of that, labeled DNA plate. So that's where you'd put your DNA sample plates. They would down stack and move to the right. The 384 head would transfer your samples from the plate back into the array tape. So you can think of the array tape continually moving along the back of that system, and it's really your assay plate, if you want to think about it that way. So going right to left, the next section or slice of the system is labeled array plates, assay plates, and that has the assay mix in it. So typically, the traditional PCR, that would have the primers and probes, custom mix, everything that you add to your sample. The array tape is then covered at the cover seal module, and then it's wound back up. There are other modules that can go in there to make it specific to the assay being run. This is just to show the modularity of the system and why that's important. So if the automation is working on your screen, then you can see that each of these is their own unit, and you can see how they separate and come back together as the whole system. So just quickly a little bit about the technology. So at Douglas Scientific, we are a technology innovator, so we've developed our own jet dispense module, and this goes down to 250 nanoliters, and we're getting 7% CV at that volume. And that is one of the things that allows us to drive costs down and keep speed up, and also use fewer of your precious samples. And this is something that gets to that better, faster, cheaper paradigm. We also, as in the partnership with EnviroLogix, we have a partnership with SciBio for their 384 heads. So when there's a tool like that available and it already works well, we didn't think there was a need to reinvent the wheel. So we're getting at 1 microliter, 2% CVs, and typical operating volume for us on the 384 head is about 800 nanoliters, and we get very consistent results at that volume. So the point there is our system is an open platform where if we think the industry needs a particular technology, we can develop that and put it on the system. If the market demands an existing technology, we can integrate that as well. So it's very flexible as a platform. So on to the applications, and more to the point of what we're talking about here today, I want to build a little bit on what Dan was talking about and just go at it from a workflow point of view. So with traditional PCR, as you're looking at across the top of the slide, it's a discontinuous process requiring multiple instruments. So you have to do some kind of plate setup. This requires some type of robotic instrument, some type of liquid handler, and then you have to do your thermal cycling, and then you have to do your detection. And there's units out there that do the thermal cycling and detection in one unit, but at the end of the day, you have to shuffle a lot of plates around, and sometimes you need additional robotics to move plates from unit to unit if you want it to be fully off. The system that we're showing you here today, as you can see along the bottom of the slide, the isothermal DNA amplification technology from EnviroLogix, running on the Douglas Scientific array tape platform, it's truly a total solution, and it's truly a walk-away solution. And so there's a lot of claims out there of total solution and walk-away, but what I want to illustrate for you here is that this is truly walk-away. You see one instrument, and it's a continuous process, and it's a single vendor solution. So also from a support point of view, we talk about pain points. Some of it's in the chemistry, some of it's in the instrument. And for those of you out there that have dealt with a lot of automation, you know that going to multiple different vendors to try to cobble a solution together, it is in itself a pain point. So this addresses that as well. So what you're looking at here is the Endpoint isothermal instrument. So this would be the instrument that all the data on that Dan sheet with you. And you can see the layout similar with where it starts with the right side of the instrument, there. And as the array tape unwinds and goes into the system, it goes into sample transfer, then it goes into the assay mix transfer, and then it's sealed. And then you see the taller unit there towards the left side, and this is an incubator. So this is what provides the isothermal environment for the reactions to take place. And this is the endpoint unit. So as soon as the wells have been exposed for a proper amount of time for the reactions to be carried out, they then go to the module directly to the left of the tall unit that you see there, and that's the detector. And so that's where you do your endpoint reading. And then after that, the array tape, you're done with it. So some customers, it goes directly into the garbage. Some customers, it's rolled up on a take-up reel there, and they'll keep it for future use. So let's just step through those modules. One is the unwind module. Two, as you see it there, is the 384 head. 3 is the plate stacker, and this is the stacker that has your DNA samples. 4 is the plate stacker that has your assay mix from EnviroLogix. 5 is the dispense jet, and this is the non-contact dispenser that puts the assay mix into the array tape that already contains the DNA sample. 6 is the tape sealer, and the reason the color is different there is because we want to emphasize that there's some temperature control already going on here. So we chill this so that the reaction is not started too early, and this really speaks to quality of data that we provide with the system. Number 7 there, that is the incubator. So that's what holds it at the isothermal temperature for the reaction to take place. And then finally, detection. Okay, on to the next slide. So isothermal endpoint nucleic acid analysis, instrumentation, consumables, software, chemistry, these are all the components. And we've looked at now the instrumentation, I've talked a little bit about the consumables. The software we won't go into today, but it's a graphical user interface. It's drag and drop. It's very intuitive. And the chemistry on the right, Dan has talked to you a little bit about. The point of this slide is just to show that it's all been brought together, again, to emphasize the total solution there. So I want to reiterate something that Dan touched on, and this is the familiar traditional qPCR reaction components versus the unique D-enabled product that EnviroLogix offers, and the difference between those two. And you're using a nicking enzyme, amplification in 10 minutes. That's profound if you've worked with traditional thermal cycling PCR and you know the amount of time that it takes to get an answer there. So I want to go back and illustrate not only is it faster, but when you look at it in the context of the actual work that you have to do in a day in the lab, again, that Dan already talked about. I want to put it in a little bit different perspective and actually quantify some of the numbers that we're claiming here so that you can see them clearly. We're talking about virtually the same number of plates, and this is in an 8-hour day, 300 versus 320. 60 days, right? So working days, that's something like 2 months, and you've got 1 day for the D-enabled Nexar. That's profound. A 60-fold difference. So when we talk about doing things better, faster, cheaper, hopefully this illustrates for you that this is really a true statement, and it really is better, faster, and cheaper. So the reaction volume also, 10 microliter total reaction volume is typical, and that's what Dan runs in this is in a fully optimized lab with really, well optimized assays down to 10 microliters. And starting point for us is 1.6 microliters. So you can see there's already greater than a 6-fold savings in the chemistry and the volume that we're using there. So on to another application, and we're really excited about this one because this is the same technology, but now for real time. And what this allows you to do is to still run the isothermal chemistry, but instead of having it go through the isothermal chamber and then out to another unit that is the reader, we've combined those two. So what you're looking at on this slide is the detection having been built into the temperature control unit. And so that's what gives you the ability to do the real-time reading. And let's just step through quickly here the modules. Again, starting on the right, you've got the unwind module, that's the same. 384 head, also the same. Plate stacker, that's for your source plate. Second plate stacker, that's for your assay mix. The dispense jet puts the assay mix into the array tape. The tape sealer is right before unit number 7. And that, again, is the detection and the amplification. And so you can see it's a combined unit. And again, what gives you the ability to use this technology to get real-time results. Okay, now this is a movie that's going to show the system in action. And there's audio on it, so I won't speak over that. I'll wait till the end.
The Nexar Optimized for real-time nucleic acid processing. Quantitative results in as little as 20 minutes. With high specificity, repeatable results, and walk-away operation, it's the solution for high-throughput genetic analysis. Now, you'll have the flexibility to run isothermal and qPCR chemistries on the same instrument solution, utilizing purified and crude sample matrices while significantly reducing chemistry costs with small reactions volumes of 1.6 microliters and high-throughput sample processing. And with 10-minute isothermal amplification time, labs go from sample to answer in 20 minutes or less. This system integrates liquid handling, sealing, thermocycling, real-time detection, and data analysis in an inline system. The process is simple. Stamp the DNA sample in 384 wells at one time using the SciBio contact dispense pipette. The array tape is automatically advanced to the non-contact dispense jet to add the reagents. During the sealing process, the assay is chilled to 20 degrees within seconds to prevent uncontrolled reactions. Samples then enter the thermocycling and detection module. Isothermal DNA amplification and detection take place in 10 minutes or less at a constant temperature of 56 degrees. For PCR chemistries, thermocycling and detection are defined by your protocols. The Intellix software suite analyzes and plots data in real time, allowing the user to verify reaction success and view results throughout the process run. Inline, automated, high throughput, real-time processing. It's time to do more with less. Douglas Scientific, partnering with you to make the world a better place.
Okay, so hopefully that movie gave you a clear illustration of how the inline array tape automated system works, and it just keeps the array tape moving through the system in a linear fashion, and how each step along the way there is a pipetting tool or a stacker tool or a detection tool, whatever's required for the assay, to run that step of the assay as the array tape comes down to the tool. So it's flipping the traditional hardware paradigm on its head, really. And we like how that's paired with the EnviroLogix technology that's doing the same thing from the chemistry side to bring really a total solution here with profound throughput advantages. And now just to summarize, the array tape automation results in profound savings, lower consumable costs. The array tape itself is significantly less expensive than the plates, and lower chemistry costs via miniaturized reaction volumes. We talked about that. More efficient use of your human resources. You saw in the movie the system really is a walk-away system. Once you load your sample and assay plates on there, you hit go. That's the beauty of the inline system. It runs. It just runs. It runs to completion, and it's finished. And we don't have time today, but I can give you multiple illustrations of the effect this has had in different customer labs, and I'd be happy to address those offline or in the questions section. And then from the chemistry point of view, the truly disruptive isothermal DNA amplification positioned to redefine qPCR. Sample to answer in 20 minutes or less. That's profound. Total solution, automated platform, and verified assays. And so there's a lot of vendors out there that talk about that. We're actually doing it. We're very, very excited about this. You've seen the results here. And I just want to leave you with one thought. If you haven't seen this quote before, I really like this one. But it says the future is already here. It's just not very evenly distributed. And this is a quote from William Gibson. And he's a famous author, for those of you that already know who he is. But I think that's really what we're talking about here today, is that we shouldn't think about this as something that's coming. It's something that's way out there. This is the kind of stuff that is happening today, and you've seen the data. There's already beta customers out there using this, so it's reality. It's here today, and I think this quote really captures that. So with that, I'd like to thank everybody and pass it on to Kathy.
Thanks, Craig. Before Cassie begins, let me just say that she's going to pull the two previous presentations together, no small order there. She's going to talk about combining the technologies discussed by Dan and Craig and provide experimental results. Cassie, we're ready when you are.
Thank you so much, Tamlin. It's a pleasure to be here today. As Tamlin just said, I'm talking about the combination of Douglas Scientific instrumentation with the DNA-enabled chemistry that both Craig and Dan have talked about earlier. I'll start with a quick overview of what I'll be talking about today. We're going to discuss experimental results that were generated here in our lab at Douglas Scientific using these two technologies, the DNA-enabled chemistry that Dan described and the Nexar platforms with array tape in the Douglas Scientific instrumentation as Craig described. Our experimental results are going to demonstrate a couple of different things about both the instrumentation and the chemistry. First, we'll be looking at endpoint and real-time analysis of isothermal DNA amplification reactions in array tape. We're going to delve into the quantitative and qualitative aspects of the DNA-enabled chemistry a little bit, and we're also going to look at a couple of different DNA samples that were used for the DNA-enabled amplification reactions, looking at both purified and unpurified DNA samples. First, I'm going to start with some experimental conditions that were common for all of the results that we're going to be showing here today. Our sample amplification and fluorescence detection was all completed in array tape. So once the samples were put into the array tape, it was sealed and everything happened in the same place after that. Our total reaction volumes were 1.6 microliters for all of the reactions that we did. And these were composed of equal volumes of both sample and master mix. So 800 nanoliters of sample was dispensed with the 384 tip SciBiohead that Craig talked about, and 800 nanoliters of 2X master mix was dispensed with our dispense jet that also Craig mentioned. Our Nexar was used for sample and master mix dispenses. The amplification was completed in 10 minutes or less at 56 degrees. So it was held constant for 10 minutes at 56 degrees, even though the reaction maybe didn't always take the full 10 minutes. Not all of our experimental conditions were the same. We did vary some of the conditions that we used. We used the Nexar Optimized for Endpoint isothermal DNA amplification for the qualitative, which would be your presence or absence analysis for our endpoint reactions, and we used the Nexar Optimized for real-time nucleic acid processing for the quantitative or real-time analysis. Again, we used a couple of different DNA sample sources. One was the synthetic DNA, which was the G block that Dan mentioned earlier. We had some purified genomic DNA samples, and we also had unpurified or crude prep samples that contain genomic DNA. Moving into our results and discussion. The first experiment that we did was on the DNable assay for soybean lectin gene, and we heard about this a little bit earlier. The soybean lectin gene is a common housekeeping gene in soybean assays. We looked at it in 4 different conditions, either with purified genomic DNA or the unpurified genomic DNA in our endpoint detection system, or we used purified and unpurified genomic DNA in our real-time detection system. So the first thing that we did was we ran an isothermal reaction in a standard industry-leading real-time PCR instrument. Much of the results that you've seen in this presentation so far have been generated in a real-time PCR instrument, so we duplicated that here in our lab using purified genomic DNA or that same crude prep, which was a 5-minute quick preparation. Looks like soy milk and a no-template control. With this, we were able to move on to our analysis on the array tape platforms. So our first reaction was we put the soybean samples in our Nexar Optimized for Endpoint analysis. We looked at, on the right, you can see purified DNA, in the middle, the crude prep, and on the left, our known template controls. You can see we have very clear distinction between the positive samples and negative samples, and the crude prep and the purified genomic DNA performed very similarly. We then ran the same samples with the same assay in our Nexar Optimized for real-time nucleic acid processing. On the left, you can see the purified genomic DNA. As you can see, it amplified in a very tight band. The samples performed very similar to each other. And on the right, we have the unpurified or crude prep of genomic DNA. All of the samples amplified very nicely, and they have exponential amplification curves, but they took a slightly different path to get there. Our next step was to push the limits of the DNable chemistry a little bit further. We wanted to look at the dynamic range of the DNable chemistry in array tape. So we took a DNable assay for a known human pathogen, took a purified synthetic DNA template. This was a G block that contained the sequence of the human pathogen. We made a dilution series that covered 7 orders of magnitude, and these were made with 1 to 10 dilutions going from 1 times 10 to the 10 all the way down to 1 times 10 to the 3 copies per microliter. And we amplified these in array tape in the Nexar Optimized for real-time nucleic acid processing. Here are the results that we got with our dynamic range demonstration. On the left, you can see this looks like PCR amplification curves. This is actually an isothermal reaction. We have shown here on the left 4 of the 8 standards. So this is the odd dilutions, so 1 times 10 to the 9, 7, 5, and 3. And on the right, we plotted out the regression line of these 8 samples. All 8 of them were included in the regression line analysis. And I would like to point out here that the analysis is a little bit different than what you're used to seeing, where in PCR we would look at threshold cycle analysis. Here we're looking at actual threshold time analysis, because all of the samples were run at one temperature at 56 degrees, so we're just looking at the time in seconds at which the fluorescence crossed a threshold of detection. And we got a nice R-squared value there. We were very happy with these results, and we moved on again to push the limits a little bit further. And here we wanted to look at the quantification of bacterial genomic DNA in array tape. Again, we looked at very small volumes. We used the DNable assay for CMS, so this is a bacterial that's pathogenic to plants. We used purified genomic DNA from bacteria as our DNA template. And our amplification reaction was done in the Nexar Optimized for real-time nucleic acid processing. I'll give you a little bit of the punchline ahead of time. We looked at 1 times 10 to the 4 copies per microliter, and those samples on average crossed the threshold at 159 seconds into the reaction, whereas samples containing 1 times 10 to the 2 copies per microliter crossed that fluorescence detection threshold at 179 seconds. And here we have the amplification curves that we generated. On the left, you can see a cluster of samples that came up. That's our 1 times 10 to the 4th samples. And on the right, you see a cluster of samples. That's 1 times 10 to the 2. They look close together, but that's just because the reaction is happening so quickly, and the graph goes out to a full 10 minutes. Our next experiment was we looked at the STIX1 gene that Dan mentioned earlier. Using that same DNable reaction for the STIX1 gene, we used 2 different strains of E. coli to look at this. We used 1 times 10 to the 2 copies per microliter of genomic DNA from 2 strains. It was 3C and 0157. We used both of our Nexar platforms, the Nexar Optimized for real-time nucleic acid processing and the Nexar Optimized for Endpoint isothermal DNA amplification. Here are the results that we got for this analysis of the STIX1 assay in E. coli. On the left, you can see the real-time DNA amplification curves. Both of the 2 strains of E. coli came up at the same time. You can see the curves are right on top of each other, even though it's 2 different strains with the same copy number going in. On the right, you can see the endpoint analysis of the genomic DNA from the 2 E. coli strains. Again, we see a very nice separation of our positive samples and our no template controls. And this experiment was done as kind of a real-life example of when these 2 instruments would be used together. For example, if you were screening a large number of samples, you might start with the endpoint analysis to look for positive or negative samples. And if a sample came up positive, you might move over to the real-time amplification to quantify how much bacteria was actually in that sample. Going on to some summary and conclusions. We're going to go all the way back up to Dan's presentation at the beginning, looking back at everything we've learned about DNable isothermal DNA amplification chemistry here today. It's a sequence-specific amplification that occurs in 10 minutes or less. The reactions occur at one temperature, in this case 56 degrees Celsius. We use fluorescent dye-labeled molecular beacon to assist in detection. We have the quantification ability using real-time detection and qualification or presence-absence analysis with endpoint detection. And this chemistry is compatible with small reaction volumes, and I'll point out also with crude preparations of your DNA samples. Going back to what Craig was talking about, the Douglas Scientific Array Tape platform, we have 2 Nexars that are both optimized for use with isothermal DNA amplification chemistry. The first is optimized for endpoint analysis, and that would be our qualification or presence-absence analysis. The Nexar Optimized for real-time nucleic acid processing is designed for real-time or quantification of samples. Both of these have inline processing, and they're designed for high-throughput sample analysis. We have miniaturized reactions, so 800 nanoliters of sample and 800 nanoliters of master mix is our standard reaction volume, and both are designed to have walk-away time from the operator. And when we combine these two technologies, we get a very powerful new solution. We get PCR-like quality results in 10 minutes at 56 degrees with our low reaction volume, so we demonstrated 1.6 microliters. We were able to get these type of results with purified or unpurified DNA. We showed quantitative analysis over a very wide dynamic range and qualitative analysis where we were looking at presence or absence with our endpoint detection. Both the chemistry and the instrumentation are very versatile and highly customizable for solutions for many scientific applications. So thank you so much for your time, and we look forward to hearing questions from you.
Thank you, Cassie. Before we start the Q&A session, I just want to give one final call on the questions for our panelists. Please submit your questions now. I also want to let you know that a post-webinar survey will deploy shortly in the presentation manager. Please take a few minutes and respond. All right, we're ready to start the Q&A discussion. We have a lot of good questions already, and we'll answer as many as we can.
First, let me say that we have way too many questions for Dan to answer in the time that we have remaining. He has said he'll respond to as many offline as he can. So the first question, Dan, that I have for you right now is, have you tried amplification in the crude blood sample?
Hi, Tamla. Yes, we absolutely have. It's amazing how resistant this assay is to inhibition by things that are going to inhibit PCR. We've done that in a variety of blood types, particularly avian at this point in some human bloods, and yes, it's resistant to the usual things that are inhibiting PCR.
Okay, then someone wants to know, can you multiplex the reaction and to what degree?
Yeah, as I showed in the presentation, I was showing duplex reactions, and those were true duplex, where you're looking at a gene of interest and a internal control of some sort, like the example of the GMO plus lectin. Right now, duplexing is certainly doable, and we've been working on triplexing, and the context there, of course, means, as with qPCR, that those are reaction conditions that are quite challenging, and you've got to work out those conditions appropriately. But there's nothing fundamentally that says that you cannot do more than a duplex. We have done triplexing. So beyond that is going to be challenging even for a PCR. Again, in the context of any sort of amplification system, in theory, you should be able to do more. In practice, those are challenging, and we're certainly working on them.
Okay, Dan, here's another one. What is the size of the Amplicon that you work with usually?
Yeah, typically from past history with qPCR, the best place to be would be in the neighborhood of like a 75mer for a TACMan type of assay. We're pretty close to that. We're a little bit smaller. It's usually in the neighborhood of like 50 to 60 base pairs. Something that, of course, it gives you the capability for the molecular beacon to, of course, anneal to the specific amplified region in between the primers and give you good specific detection of that particular amplicon. So on average, yeah, in the 50 to 60 base pair range.
Okay, Craig, I have a question for you. Detection of fluorescence, is it laser or CCD based?
Yeah, actually it's LED excitation and PMT detection.
Okay, that was quick. Cassie, I have a question for you. I'm going to have a hard time reading it. Can you talk about the throughput potential for the Douglas Scientific Endpoint and real-time instruments?
Yes, thank you, Tamlin. So our Nexar Optimized for Endpoint isothermal DNA amplification analysis, it has a throughput of about 147,000 data points per day, and the real-time instrument has about 16,000 samples per day throughput. And that's with a 10-minute incubation reaction.
All right, Cassie, I have another one for you. Can any of these experiments be done on a regular thermocyclers, or it is a Nexar-only technique for now?
They can definitely be done on a standard real-time PCR instrument. It just needs to be programmed to hold a constant temperature and detect in real time, and that can be done with some of the programming software.
Okay, unfortunately, that's all the questions we have time for now. As I said at the beginning of the Q&A, our presenters have said they will answer as many questions as they can offline. We have your email addresses, and we'll take care of that. Thank you very much for joining us today. Thanks for listening in, and thanks, presenters, for the great presentations. This webinar will be archived for 6 months on our website, www.genengnews.com. If you missed parts of it, you can watch it again, or you can recommend it to friends and colleagues. Once again, I want to thank all of our panelists as well as our sponsor, Douglas Scientific.